In 2018, the adult human neurogenesis debate came to a head when two important studies disagreed about whether new neurons form in the adult human hippocampus. This argument became a key moment in neuroscience because it was a fight between the hope that neurogenesis exists and the doubt that it does. Boldrini's group published a study in 2018 that showed hippocampal neurogenesis even in older people. The article suggested that healthy older people keep their cognitive and emotional integrity because their hippocampus keeps making new neurons. However, in the same year, a study led by Alvarez-Buylla, Huang and Garcia-Verdugo published findings in Nature, reporting that neurogenesis was undetectable in the hippocampus of adult human brains and had already dropped sharply by the time a person was 1 year old compared to when they were born. So, what might have led the labs to reach different conclusions on the same matter? We are making a series of videos that will look at the subject of adult neurogenesis over the course of several episodes. First, we'll talk about the history of neurogenesis discovery and how scientists found it in human. Then, we'll discuss the 2018 debate about whether neurogenesis exists, which sparked a new interest in the field around the world. The discovery of adult neurogenesis. Challenging brain dogma. Let's talk about the history of neurogenesis to start the story. So, what is adult neurogenesis? Adult neurogenesis is the process by which new neurons are made in the adult brain, mostly in the hippocampus, which is important for memory, learning, and controlling emotions. For an extended period, neuroscientists believed that the adult mammalian brain remained fixed and non-regenerative, with no formation of new neurons after birth. But in 1965, Altman and Das published a study in the Journal of Comparative Neurology that went against this common belief. They injected rats with a radioactive form of thymidine and used it to label newly synthesized DNA in cells. Thymidine is incorporated into the DNA of dividing cells during replication, allowing researchers to track cell division over time. Altman and Das looked at the hippocampus in particular and found thymidine-labeled cells in the dentate gyrus of the hippocampus in young rats, although the number declined in older rats. Their research provided the first direct evidence of adult neurogenesis in mammals, which goes against the belief that the adult brain cannot generate new neurons after birth. In 1969, Altman also found the subependymal layer of the lateral ventricle to be another neurogenic niche. He observed labeled cells proliferating there and migrating to the olfactory bulb. Expanding the understanding of adult neurogenesis from studies in songbirds. After Altman and Das's studies, Nottbaum and his colleagues at Rockefeller University studied songbirds to explore brain plasticity in relation to song production. They discovered the presence of new neurons in the vocal control nucleus, a brain region involved in song production. This discovery was important because it suggests adult neurogenesis is functionally significant. Connecting the formation of new neurons to song production. Environmental enrichment enhances adult neurogenesis. Later, studies also began to show evidence that brain plasticity is linked to external factors, suggesting that adult neurogenesis can be boosted by environmental enrichment. One of the famous studies is from Kemperman's group. In their study, they found more hippocampal neurons in adult mice living in an enriched environment. If you don't know, enriched environments usually means larger cages. There is also social enrichment, which may involve housing more than one animal together and providing them with various sensory stimuli, such as running wheels and toys. Kemperman's study put adult mice in either standard cages or enriched environments. Mice that lived in enriched environment had 15% more granule cell neurons in the dentate gyrus of the hippocampus than mice that lived in standard cages. First evidence of adult human hippocampal neurogenesis. So far, scientists have been able to show that adult neurogenesis happens in animals. Altman and Das showed it in rats. Nottbaum and his colleagues demonstrated it in songbirds and proposed its connection to crucial functions. Kemperman also found that external factors can affect neurogenesis in mice. But there was still no proof that neurogenesis happens in human. In 1998, Eriksson in Sweden published results in Nature Medicine. The study was a big deal in neuroscience because it provided the first direct evidence of adult hippocampal neurogenesis in human. Coming up next. Next, we'll look more closely at Eriksson's study of neurogenesis in human. Also, we will talk about how the results of neurogenesis research became more and more mixed as it went on. The findings even brought up an important question. Do adult humans really grow new brain cells in the same way that animals do? So, what do you think? Do you think adult human brains are really capable of growing new neurons? If you enjoy this type of video content, don't forget to subscribe to our channel. Stay tuned for more.